So if you joined us today to learn about gene therapy and rare disease, you've arrived at the right place. Our board of directors and all of us at the International Rett Syndrome Foundation want to thank you for being with us today. I'm Paige Nuez, Director of Family Empowerment for the Foundation. And all of us at the Foundation want to thank you for your support and engagement, because continuous awareness and empowerment is critical to our shared success. And I say that both as a parent to a beautiful teenager with Rett syndrome and as your advocate at IRSF. We want you to be as educated and empowered as possible as you care for your child and as you speak to others about Rett syndrome, and for the professionals who have joined us today as you work in your field of interest. So welcome and thank you to all of the families, caregivers, clinicians, researchers, and industry for being with us today. We believe that information on our rare disorder should be free to all who avail themselves of it, and we want to thank our donors and supporters for that shared belief. Our 2021 educational programming is made possible through the generous support of Acadia, Novartis, and Taysha Gene Therapies. We're grateful for their support, which allows us to connect and empower you to this important information while we invest further in our research programs. Speaking of research programs, we have some QR codes on the next couple of slides. So please pull out your smartphones, open your cameras, and scan these QR codes to take you to some really important online information. The first one I want to share information with you about is the Rett Research Syndrome Registry. It's part of IRSF's proprietary Rett Research Program. And the new registry is a significant way that you can participate in the research process. The NIH NCATS RDCRN funded natural history study is ending this month. And because of this, over the last year, Dr. Prashad and others have worked really closely with the PI doctors at the US Rett clinics to design a registry that will take the place of the natural history study dataset. Later this year, your local IRSF Center of Excellence will offer you the opportunity to register for the Rett Syndrome Registry. And this will allow your loved one's data to be part of the growing repository of information that's changing the landscape of drug development. So every caregiver and individual with Rett syndrome has the opportunity to be an active research partner in this mutual goal of identifying treatments and cures for our loved ones with Rett syndrome. And participating in the Rett Syndrome Registry is a really important step. So please go ahead and scan the QR code or visit our website, rettsyndrome.org, for more information when this registry becomes available. Also, it's back to school time. So as we face back to school and many day programs and therapies are beginning to happen in person, we want you to be as empowered as possible to help those working with your children to advance communication. So we encourage you to buy the evidence-based, research-supported communication guidelines in Rett syndrome to help everybody work with your loved one on how to advance communication. Next announcement I have is a request. We have another request of you to please contact your senators to fund Rett research. As the Senate has yet to consider the FY22 appropriations bill, we urge you to reach out to your US senators and request their support to list Rett syndrome as a topic eligible for funding through the Department of Defense PRMRP program in the defense funding bill. Your advocacy has helped secure upwards of $8 million for Rett syndrome research in recent years, which is directly equates to progress that we all are hoping for and want to see for our loved ones with Rett syndrome. So your efforts really do matter. Again, scan the QR code and we've created easy access for you to find information on writing to your senators. Last announcement is we're gearing up for Ascend 2022, Rett Syndrome National Summit in Nashville, Tennessee. First opportunity for us to be in person in a really long time. We're looking forward to seeing you there. And a reminder that early bird registration pricing is open through September 30th. So please get online, visit rettsyndrome.org, start making your planning and take advantage of that early bird pricing. All right, so with those announcements, thank you. I'd like to get on to today's RettEd topic. We receive a lot of questions around gene therapy and gene replacement therapy, specifically in Rett syndrome and its potential for single gene disorders like Rett syndrome. IRSF's Chief Science Officer, Dr. Dominique Pichard, is going to speak today with some expert panelists to unpack what's involved in delivering complex treatment molecules to the affected cell, learn what NCATS is doing to accelerate clinical trials in rare diseases, and allow us to speak with Dr. Jill Jarecki, who is the Chief Science Officer for CureSMA, who's going to share their lessons learned from their FDA-approved gene therapy clinical trial experience. And throughout the course of the presentation today, we'll be answering the questions that you pre-submitted at the time of your registration. So again, thank you for being with us, and I am going to go ahead and turn things over to Dr. Pichard.
I switch over to phone. So I was saying, today we're going to talk about gene therapy, as Paige had introduced. And the Rett syndrome community is certainly no stranger to the term gene therapy, as it's been discussed as a potential therapeutic modality for Rett syndrome for more than 10 years. And since there's been a tremendous amount of research conducted, and the most exciting part for our community is that this has culminated in 2 biopharma companies with clinical programs in development. These 2 companies, Novartis Gene Therapies and Taysha Gene Therapies, have publicly announced that they are working on submitting an investigational new drug application to the FDA by the end of 2021. So with the potential of having gene replacement therapy trials in the near future, it is so important for our community to learn about and understand gene therapies better, in particular the gene replacement therapy, so that when there is an opportunity to consider this therapy for your loved one, you're informed, and therefore you feel like you can make an informed decision about the potential risks, the potential benefits, and decide if this is the right approach for your loved one with Rett syndrome. I want to start off by introducing our 2 guests, Dr. Jill Jarecki and Dr. PJ Brooks. Thank you both for joining us today to share your knowledge in the gene therapy space. Our conversation is centered around what are the challenges with developing gene replacement therapy? What are the potential risks to patients who receive gene therapy? What has been the experience in the SMA community before, during, and after the clinical trial? And now that there's an FDA-approved gene replacement therapy for SMA. I'm going to start off by introducing Dr. Jill Jarecki. Dr. Jarecki is the Chief Science Officer of CureSMA, a nonprofit foundation dedicated to creating a world without spinal muscular atrophy and supporting families living with the disease. Dr. Jarecki has managed a research portfolio of over $55 million in research investments. The SMA community has the great fortune to have 3 therapies FDA approved for SMA. So before I turn over to introduce Dr. Brooks, I wanted to see if Dr. Jarecki, you could tell us what is SMA?
So SMA stands for spinal muscular atrophy. And SMA is a neurodegenerative disease that primarily affects motor neurons and perhaps other tissues as well in the most severe form of the disease. It is an autosomal recessive disease, which is caused by mutations, primarily deletion of a gene called the SMN1 gene, or the survival of motor neuron 1 gene. There is a unique genetics where we have a backup gene called SMN2, which is partially functional. And SMN2 has been the basis of a number of 2 of our approved drugs, and that Dr. Pichard mentioned. One of the interesting things about SMA, which I think will be relevant to this discussion that we're having, is that SMA has a wide range of severity. We particularly traditionally have a severe form of the disease, which about 50% of the patients have, where they are diagnosed under 6 months of age, will typically be on permanent ventilation or have passed away at by 2 years of age, with a median survival of 8.5 to 10.5 months. And these patients primarily have 2 copies of the SMN2 gene, although that can vary a little bit. We have an intermediate form of the disease, or where the patients present over 6 months of age, and those patients can sit but never walk, and we call that SMA type 2. And those patients primarily have 3 copies of the SMN2 gene. And then we have a mild form of the disease where the patients are ambulatory, but typically lose the ability to walk sometime in their lives, and they present over 18 months of age and have more than 3 copies of SMN2 and a very rare adult onset form. SMA does not typically affect cognition. As I said, it affects motor neurons, and one of the interesting points that's probably different than in this community situation is that motor neurons are lost in SMA, and once you lose motor neurons, it's very difficult to get them back, and this creates a critical window or a period where treatment is really important, and after that you will get less impact. We have 3 approved drugs. One is a gene replacement therapy called Zolgensma, and then we have 2 other approved drugs called Evrysdi and Spinraza. Both of those drugs address the backup gene and cause the backup gene to function better by correcting a splicing defect there. Zolgensma is approved for only children under 2 years of age, where the other 2 drugs are approved for all ages and stages of SMA.
Thank you, Dr. Jarecki. Thanks for giving us that overview of SMA and the heterogeneity and the different types. So now I want to introduce Dr. PJ Brooks. Dr. Brooks is a program director at the National Center for Advancing Translational Science, or NCATS, in the Office of Rare Diseases Research. This is at the National Institutes of Health. While at NIH, Dr. Brooks has served on the Trans NIH Gene Therapy Working Group and the Regenerative Medicine Innovation Project. One effort that Dr. Brooks is heavily involved in is the implementation of the Platform Vector Gene Therapy Pilot Project called PAVE-GT. This program aims to make gene therapy development and clinical testing more streamlined, more efficient, and potentially more accessible to many patients with rare diseases. Dr. Brooks, can you start off by telling us a little bit about the PAVE-GT program, including even what financial investment NIH has made into this project and why NCATS is investing in this initiative at a time that there are pharmaceutical companies and foundations that are also investing similarly in gene therapy?
Sure, happy to. And first of all, thank you for the opportunity to speak here. I can tell you about PAVE-GT, which stands for Platform Vector Gene Therapy, but it might be easier if you can pull up the slide and I can show a little bit about what we mean by a platform. So yeah, so this is from our website actually. And so when we talk about gene therapy or AAV gene therapy, it's often referred to as viral vector gene therapy. So AAV stands for adeno-associated virus. It's a small virus that many of us are exposed to that doesn't cause any human disease. And so what you do to make a gene therapy using AAV, as illustrated on the top of this illustration, is you basically take the viral genes out of the outside of the virus, which you call the capsid, and you put in the therapeutic genes for in normal copies of the genes that are missing in different diseases. And you can see how it's a platform, because depending on what genes you put in there, you can make a therapeutic for disease 1, disease 2, disease all the way up to X. And then to think about what these viral vectors do, we tend to think of them as a good analogy as like a delivery box, like a FedEx box that's pre-addressed to go to specific cells and tissues, like cells in the muscle and the liver and the brain. In fact, the one in PAVE-GT we're using is called AAV9 that goes to a lot of different tissues. And so basically by swapping out the therapeutic genes you put into these delivery boxes, you can make therapies, in principle at least, for different diseases fairly easily. And so the idea of the PAVE-GT, this platform vector gene therapy project, has to do with the reality of how difficult it is to start up a clinical trial for each one of these diseases. And typically when people do this, they develop these things for one disease at a time. And they develop the vector and the drug, and then they have to test where it goes. They have to test the toxicity. They have to do a lot of tests to be sure it's safe and effective. But our thought was, well, if these really are a platform and all you're doing is swapping out the therapeutic genes, maybe you don't have to do each one of these steps each time over and over again. If you've done it once, maybe that would be enough. And we wanted to find that out. And so to do that, we've decided to do this sort of experiment, which is to go forward with gene therapy trials for 4 different rare diseases, all of which are under study at the NIH Clinical Center. And all these diseases are, or at least at the time we started, there's no commercial interest in any of them, because we're particularly focused on diseases that are so rare that no company is going to do this on their own. And the parents are ultimately going to have to find funding for it. So really thinking, how do we make it easier for them? How can we make the process easier? So that's the idea of the PAVE-GT program, is to go forward with these 4 gene therapies, but while we're doing it, make all the data publicly available as we go, including the INDs, which are the approvals to do the clinical trial, and all of our discussions with the FDA about how much we can streamline the process or not, to make all those questions and the FDA's answers public and put them on a public website. So that helps people to see what we're doing and the efforts to streamline the process and make it all transparent. And part of the reason we're doing this at the NIH Clinical Center, in addition to having some amazing clinicians who study rare genetic diseases and are perfectly suited to do these trials, we wanted to be able to put all this into public domain. And if we had done this, obviously with a private company, they would never allow that. And if we had done it at a university, there may be issues with intellectual property. And by doing it all within the government, we could avoid all that, and with the goal of making it all public. So that's the basic idea of PAVE-GT. We're doing the diseases are 2 metabolic diseases that primarily affect the liver, and 2 neuromuscular junction diseases that really affect the communication between the spinal cord and the muscles. And then why did NIH decide to do this? It really does have to do with this question of what do we do for diseases that are too rare to get any commercial involvement. And so ultimately it often ends up that the families have to try to support these efforts. And it's a hard thing to imagine that that's the situation we're in, but that is the reality, that you could imagine, and indeed scientists understand diseases well enough that we could develop an effective treatment using AAV gene therapy for diseases that are very, very serious. And it doesn't happen not because of a lack of scientific knowledge, but because the diseases are just too rare. And we're by trying to streamline the process and benefit all those families, and indeed the whole field, we feel like that's well worth the NIH investment. And we're very excited about doing this. And I should also mention that it's not just NCATS. We're working with the clinical investigators from the Human Genome Institute, Chuck Venditti and his colleagues, and NINDS, Carson Bonham and his colleagues. So it's a group effort, including many of my other colleagues at NCATS as well. And we can circulate the website, but I think I'll stop there and go on to some other questions or whatever you'd like to do.
Thanks, Dr. Brooks. Is there anything different about a gene replacement therapy compared to, let's say, when you develop a blood pressure medicine or something like monoclonal antibodies? Is there anything different about the gene therapies in terms of like the manufacturing that it's helpful to have this kind of what will be created this booklet that someone else can look at, the plug and chug. Is there utility in that? And then why is it different than other types of drugs?
Well, I think there's a lot of differences. I mean, the one fundamental one is the difference between a gene therapy and what we call a small molecule, like a typical drug. And so one of the things, even at a basic safety level, when you're developing a brand new small molecule, you can often do the safety test on normal human volunteers. They take the drug and you basically see if they get sick or not. You cannot do that with the gene therapy. You're putting in an AAV vector into people, and once you put it in, it's never going to come out. So that's a permanent modification. You can't do a standard safety testing, a phase 1. You just would never do it for ethical reasons. Another one is that it costs a large amount of money to make these drugs, recombinant AAV vectors, typically in the millions of dollars each time you do it. And so they're very expensive. They're regulated by a different part of the FDA, the Center for Biologics, as opposed to the Center for Drugs. And of course, as I said, the other thing is there's many people in the world that have high blood pressure, but we're talking about diseases that may affect less than 100 people and may never be commercialized. So there's many differences.
Thank you. And what type of investment is this costing NIH? Are we talking a few $100,000? Are we talking, what type of investment is NIH making?
No, we're probably for PAVE GT, it's going to be between 30 and 40 million, because we're talking about making the vectors for all 4 of these and carrying out the clinical trials, and also all of the toxicity testing that is required, and the various studies that the FDA requires to do this. And part of what we're trying to do, though, is hope to test whether some of that, if we're going to just use the same vector and just swap out the therapeutic gene and make it in the same place, the outside of the delivery box should be the same. That's what the cell sees. That it doesn't see what's inside. That maybe we can avoid or skip some of these steps, and that would ultimately save money and save time. So in a sense, we're spending money to see if we can save money and time later, and not just us, but all of the patient groups, and indeed others, everybody who's going to be developing gene therapy. We're going to make the material available so that everybody can use it, including people, companies as well.
Excellent. And it sounds like it's a very worthwhile investment that hopefully will impact people far beyond the 4 conditions that are in this initial pilot project.
Yeah, I just say, I think the point is it really is a broader experiment in how to streamline the whole process. We could have chosen 4 other diseases and done the same thing. The choice of diseases was partly based on the disease or under study at the NIH and various other reasons that we thought these 4 would be well suited, but it could have been, other 4. That's kind of the whole point.
Yeah, great. Thank you. So now let's talk about some of the clinical aspects of gene therapy. Dr. Jarecki, can you tell us about the clinical trial for gene therapy in SMA that ultimately led to the approval of Zolgensma?
Sure. So there were actually 3 different clinical trials done in the US, as well as 2 more done globally. The first clinical trial started in 2014, and the clinical trial for Zolgensma, which is the gene replacement therapy for SMA, was done in a subset of the SMA patient population. If you remember earlier when I described the SMA community, we have about 8,000 living patients with SMA, and about 300 to 350 patients born every year in the US. And so the first clinical trial, all of the clinical trials required a confirmed genetic diagnosis of SMA, and that diagnosis was specific in that it had to be a homozygous deletion of the SMN1 gene. We have 5% of our patients that have point mutations, and they were not eligible for these trials. The first trial, as I said, was a phase 1 trial, and it was started in April of 2014. It had 15 patients. There was 3 patients in a low dose and 3 patients in a higher dose, which was called a therapeutic dose. These patients had to be under 9 months of age and had to have 2 copies of the SMN2 gene. So there were quite a few inclusion criteria that restricted the number who could be in this clinical trial. This clinical trial occurred at one site in the US, Nationwide Children's Hospital in Columbus, Ohio. And the primary intent of that trial was safety, although there were secondary efficacy endpoints put into the trial, which were primarily a combined endpoint of either reaching death or permanent ventilation, as described as needing respiratory support more than 16 hours a day, and then also motor milestone endpoints of being able to sit. In the most severe support type of SMA, the definition of it is that the child will never have the ability to sit. The very positive results of this trial led to a pivotal or registration trial, which began in 2017. It had 22 participants, 16 sites in the US. And again, these children had to be under 9 months of age and have 1 or 2 copies of SMN2. You know, one trial wasn't enough. We had to do 2 trials to have FDA approval. I think this is for a few reasons. One, it's important, I think, to do a multi-site trial, not just a single-site trial. Also, to get FDA approval, you have to be using the same material or your commercial manufacturing process, and that was not the case for the phase 1 trial. So there was a different source and manufacturing process being used for this phase 3 trial. We then also have a third trial, which is to look at, in the most simplified way to think about this, is pre-symptomatic treatment of SMA for babies under 6 weeks of age, less than 42 days old, who had 2 or 3 copies of SMN2. So like I said, in SMA, once you lose motor neurons, you can't get them back. So trying to see what the difference in outcome would be between symptomatic and pre-symptomatic infants in this trial. So 3 US trials with a total of about 70 patients in all 3 trials.
Thank you. That's not, you brought up a lot of great points that I kind of want to dive into, and Dr. Brooks, I'll ask for your, you know, what you can weigh in on based on the platform, the paid GT that you guys are planning. So one thing that you mentioned, Dr. Jarecki, was that there was a requirement for genetic testing to be done before you have to have a certain type of mutation. Now, one question that we got from a registrant is, do you need to do whole genome testing in order to qualify for the gene replacement therapy trial that you know, in the SMA trial?
No, so you did not have to do that, and that is because they required a very specific mutation, which was deletion of the SMN1 gene, and we have a diagnostic test that can look for the presence or the absence of the SMN1 gene, so there was no need to do whole genome sequencing. Now that may be a very different situation in the Rett population. You know, we have a pretty unique situation in SMA where 95% of our patients have the same mutation. So.
And Dr. Brooks, do you anticipate requiring whole genome sequencing in any of your clinical protocols, or will it be more gene-specific testing to confirm they have a mutation?
We anticipate it would be gene-specific. because I don't see any reason why you would need whole genome. I think the gene specific one is really important though, because for all of these diseases, we're talking about recessive diseases where there's a mutant gene and you need to put in a normal copy of that gene. And so it's really, really important that you know that the patient has the mutation in the gene that you're putting back. You really can't screw that up. And there are diseases where the clinical presentation, like some of the ones we're looking at, the clinical presentation looking almost the same. But if it's from a mutation in a different gene, our gene therapy will not work. So I'm certain we'll need to do gene-specific testing, but I can't see any reason why we would need to do whole genome.
Excellent. And along the lines of kind of what types of mutations, Dr. Jarecki, you commented how in SMA, 95% of your patients have the same mutation. In Rett syndrome, we have hundreds of different mutations on the same gene, and there are different types of mutations. Dr. Brooks, do you anticipate that for a gene replacement therapy, it would matter if you have a single nucleotide deletion or a large deletion, or you have a missense mutation?
Yeah, I think it might for a couple reasons. One sort of has to do with the immune system and how it works and how it reacts. So if you have a person and they have say a complete gene deletion, then their immune system will never have seen the normal copy of that protein. And so if the gene therapy works, there's a chance that the human immune system will recognize the therapeutic protein as foreign and launch an immune response against it. Whereas some cases, like in many of the missense mutations, there is actually a protein made, it just doesn't work. And in that case, the immune system is probably used to it, so to speak, and sees it as self and is less likely to respond with an immune response. That could be a problem. So I think that distinction is important and is one that we'll take into account as we think about the immune modulation part of AAV gene therapy, which is increasingly standard. And I guess the only other thing that's worth mentioning as you think about different types of mutations is that depending on the mutation, some of them may be amenable to other therapeutic strategies as opposed to, nonsense mutations, for example. There are other therapeutic strategies that could be applied for or available for patients that have nonsense mutations or even certain types of splicing mutations.
Excellent point. And one thing for the Rett syndrome community, the gene of interest is on the X chromosome, and the majority of our individuals with Rett syndrome are females. And so for females with Rett syndrome, they do have one active, what we call wild type or normally functioning MECP2 gene. And so the body has recognized MECP2 already, and therefore that immune response for a female who has an active, who has two copies, only one with a mutation, that would be less likely to be a concern. But you bring up immunomodulation, and I'm wondering, Dr. Jarecki, was there a need to take what we call immunosuppressive medications or things that kind of stop your immune system? And if so, what are those immune suppressants, and how long do your patients have to stay on that?
Yes, so this was discovered in the phase 1 trial after the first few patients were dosed, is that there is the potential for liver injury and liver inflammation due to the gene transfer. And so patients start receiving prednisone 1 day prior to the infusion, and they continue on it for at least 30 days. And liver function needs to be monitored for up to 3 months after infusion, according to the FDA label. And the patients may need to be on the immunosuppressant for more than a month, depending on the situation with their liver enzymes and tracking those. But that is standard protocol now for use of this gene replacement therapy.
Dr. Brooks, as the PAVE-GT program kind of is in development, has there been consideration of other types of immune modulation or other immunosuppressants, or is kind of the prednisone that Jarecki talked about going to be standard?
I think there has been discussion and it's ongoing. It's really, those are discussions primarily from the clinicians who are involved in this and are much more knowledgeable than me about the specifics. But I do think we'd like to have a standard immunomodulation. We'd like to standardize everything. And it may well be something more than just prednisone, particularly in cases where the mutation is such that it is likely that the person's immune system has never seen the protein. You may need to do more, stronger, shall we say, types of immune suppression. But I will leave that up to my clinical colleagues who know what they're doing to figure that out.
Yeah. Absolutely. Yeah, and so it seems like there's no yet one standard protocol that's going to work for all gene therapies across the board. It really is going to be something that, as these different clinical protocols get developed, take into consideration the specific patient population and and determine what is best. But overall, there certainly seems to be a need for some immunosuppression, either because of the liver injury or, as you mentioned, concern about immune response to the new protein.
Yeah, and I think it will also depend on the route of administration. If you're going directly into the brain, the concern about the liver is less. Whereas if you're doing some of these things where you inject intravenously, all of the AAVs, a lot of it goes to the liver, and we've seen issues with that. Not only immune issues, but also if there's some sort of pre-existing liver abnormality, then this large amount of virus going to the liver can be very serious.
I'm glad you brought that up. And you mentioned two different ways to give gene therapy. One, you said directly into the brain, and you said one intravenous. So I assume you mean like you're getting a needle into your arm where your veins are, and you're getting an injection there. Is that correct?
Basically, yes. Yeah, I think it's the arm, but yeah.
Somewhere that's not the brain itself.
Not the brain. Some vein.
You can tell about.
For SMA, it's an IV infusion. It takes 1 hour. It's very simple to administer.
And so IV is intravenous,
Yes.
Into a vein. Often intravenous things are in a larger vein in the arm or something, and so you just sit there for an hour. You get your infusion, and then you're done with the dosing itself. There's also been discussion of having delivery directly into the spinal canal, or into the fluid that bathes the spine, so something we call intrathecal. And so that could be delivered through what we call spinal tap or a lumbar puncture. And that could potentially also not have the same liver toxicity of the intravenous, because more directly into the fluid that bathes the brain, as opposed to circulating in your blood, which goes to your liver. And Dr. Brooks, you mentioned directly into the brain. Can you just very generally, not in detail, but what does that mean to get an injection, to get therapy into the brain?
So aside from the intrathecal that you mentioned, for certain diseases where you really want to target a specific region of the brain, what that basically means is drilling a hole or one or more holes, and through the skull, and putting a large needle, a very thin needle called a cannula, that goes down to a certain part of the brain, and then you just slowly infuse the AAV through that cannula. and that has been done. You know, disease where you might do that would be things like Parkinson's disease, and also that was what was done is with AADC deficiency, where you're just really trying to hit one part of the brain.
Great, thank you for talking about those different types. I want to go back to, there have been questions about kind of how many patients, and Dr. Jarecki, you talked about that first trial had 15 patients, but ultimately there were about 70 patients that were in those clinical trials. What was kind of the response in the community once, you know, there was knowledge that there was going to be a gene therapy clinical trial, to then learn about that it was, I'm going to say, a very small subset of the population that was eligible, and also that could ultimately do a trial. Now, what was the community response to that?
Well, I think there was great interest in gene therapy in the SMA community, but I think two things helped the community understand the rationale for this. One was that CureSMA, we had been for years explaining that the intravenous delivery of the drug would likely be for the most severe and youngest patients, and that we were trying to fund an intrathecal delivery, like we just discussed, an IT delivery program for older and larger patients, where we could bring down the viral load and put it directly into the CNS. But we also had other clinical trials going on, namely for Spinraza, which is an antisense oligonucleotide, which corrects the splicing defect in the SMN2 gene. And so there were ongoing clinical trials for a much broader range of SMA patients with Spinraza. And so those were available. And also Spinraza was approved in December of 2016. So there was another commercially available option for patients. And I think that changes, you know, although people were extremely interested in gene therapy, a one-time drug treatment, when you have other disease modifying options, it takes a lot of pressure and, you know, stress, because there are other treatments available. So from that sense, we were very lucky to have multiple different options in the SMA community available to our patients.
Absolutely. I can see that making it, you know, knowing that you have another option for your loved one makes that certainly easier. I also wanted to ask, there's been a question that's been asked to us about how many different treatment times do you have with gene therapy? So Dr. Jarecki, I'll have you start us off on answering that. Like, how many times do you get your gene therapy infusion?
So you only receive gene therapy one time, so it's a one-time treatment. I think, you know, we have questions largely in the gene therapy research community about how durable gene therapies will be and whether repeated administration could possibly be needed. But in SMA, we have a long-term follow-up study of our first clinical trial where patients have been in there for, you know, we have data from those patients for more than 6 years now, and those patients are doing very well and had no regression of the function that they obtained that, you know, was much greater than natural history. So we see no signs of loss of efficacy of the gene therapy in the SMA situation at all. I mean, I think Dr. Brooks could probably address this, but I think there are probably issues with immune response for redosing particularly systemically with the IV approach. Maybe you could do it intrathecally with the current AAV vectors that we're using. So.
Thank you. So for the SMA gene therapy, it is a one-time dose only, and what you've seen so far, there doesn't seem to be any loss of effect over the time that it has been watched so far. And Dr. Brooks, is your plan to also have a one-time dose in the PAVE-GT pilot program?
Yes, it is. And we anticipate that would be sufficient for most of the diseases and most of the patients we would be looking at.
And can you talk about reasons why you might not be able to get a second dose?
Sure. So as Dr. Jarecki mentioned, once you get the first dose of an AAV, your body does generate an immune response to the capsid part, the outside part, as well as other things. And so after that happens, you've got circulating antibodies in the blood. And so if you tried to use redose with that same vector, those antibodies would block it and prevent it from getting to where it's supposed to go. So we do think of these as a one-time shot, although I do think we're realizing different ways to modulate the immune system to make redosing possible. There's a lot of kind of interesting approaches to try to do that, various ways to modulate the immune system. And the other thing is you could hypothetically use a different AAV serotype, a different one of those delivery boxes, if you will, that the immune system doesn't recognize, and that theoretically could make redosing possible. And some situations that might be important might be if you were to treat, say, a very young baby and do gene therapy to the liver. As the baby gets older, the liver grows. Every time a liver cell divides, the AAV essentially is lost. It doesn't integrate into the genome, and that's a good thing in terms of safety. But as the cells divide, then the genes are lost. And so hypothetically, it might be possible if you treated a very young child with gene therapy to the liver, some of it might be lost as they get older. But I think we would, we'll deal with that bridge when we come to it, is my understanding of the field.
Yeah, so right now there's still maybe as many, if not more, questions and answers to some of this. But with today's technology and kind of options, it sounds like one time is all we can get because of the immune response to the virus itself, to parts of the virus. Although that may or may not be the case if you're getting the delivery intrathecally or directly into the brain, that'll maybe stay under the we don't yet know. And you mentioned that there's a lot of work being done to circumvent that antibody issue to the viruses, so that maybe in the future there could be redosing.
Yeah, about a year ago, we had a meeting at NIH that focused specifically on immune responses to AAV, and I could find the link and put it in the chat, but there's a lot of discussion about some of those newer approaches that I think people who are interested in that would find very interesting.
Great, thank you, Dr. Brooks. And Dr. Jarecki, you mentioned that in that very first trial, there were 3 individuals that were in a lower dose, and then 3 individuals that got a higher dose, and I believe you said that was then considered to be the therapeutic dose.
Mm-hmm.
Does that mean that the individuals that got the lower dose didn't benefit, or maybe didn't benefit as much?
Well, I think there was the potential for that situation. For safety reasons, they like to explore a lower dose first and then move up to a higher dose. And there was the potential that the lower dose would not have as much efficacy as the higher dose. Fortunately, both doses did have efficacy, and we saw changes in natural history in both of those cohorts in that trial. There were 3 babies in the low dose cohort and 12 babies in the high dose cohort. But that is, I think, something that the patients were carefully consented about when they joined the clinical trial.
And when you say something they were carefully consented about, just the fact that the ideal dose wasn't yet known?
This is the first time you're going into a human being and you're extrapolating the dose from animal model studies, and you may not get the dose exactly correct, right? Without, you know, and so there's always the potential that there may be a little bit difference in the dose response curve in humans than there is in the animal models, and there may need to be some adjustment of the dose, which is one of the reasons why you're doing the clinical trial.
And that's not something that's
experiment.
Go ahead.
Clinical trial is an experiment where you're trying to understand and determine a safety, efficacy, dose, particularly in those early stage clinical trials. And some of those, you know, to do your pivotal registration trial. And it may be that you will have to have adjustments on some protocols, like we had to have with adding in the immunosuppressant for a month, or, you know, dose adjustments as you move along in clinical development.
Those are great reminders, you know, that it is still experimental, and that's why it's research.
Yes.
Because we don't yet have the answers. We're trying to learn if it works and if it's safe and at what doses. And Dr. Jarecki, you talked about kind of the age range for those who can access the gene therapy, and it really, for the approved therapeutic, it's the very young individuals who have a severe form. And Dr. Brooks, for the PGT conditions that have been selected, will it also be all very, very young individuals, or are these conditions where the age span of a clinical trial, in theory, could be broader?
It could be broader, and in fact, that's one of the discussions, one of the first discussions we'll be having with the FDA, is what the appropriate age of the first patient should be. You know, there's a long history about having discussions about that. I think the general sense is the preference should always be to start with adults if possible. Obviously in SMA, the severe form, that wasn't possible, but in some of these diseases that we'll be studying, it probably will be. And I think we need to discuss with the FDA what makes the most sense, and with our clinicians who know the patient cohort to try to decide that.
Excellent. So potentially gene replacement therapy is something that can be used outside of a very small age, depending on the condition. We talked about earlier how SMA is a degenerative disorder, and so once those motor neurons are gone, there's nothing to correct. And that's not the case in all genetic disorders. Certainly Rett syndrome is one where that's not the situation. And you know, in the condition that you're looking at in the PGT program, it's also sounds like not a concern.
Yeah, none of them are degenerative. One is a liver disease, and the neuromuscular junction one is interesting because the nerve is there and intact, and the muscle cells are there and largely intact. The problem is that the two don't connect, and so we anticipate that, and there's published animal data that if you can use gene therapy to make them connect, that should have clinical benefit.
I just point out in Europe, they have a different drug approved, a label for Zolgensma, which instead of an age limitation, they have a weight limitation of, I believe it's 21 kilograms, which was roughly approximate to a 5-year-old child. So they actually have a broader label in Europe than we do in the US. And Novartis Gene Therapies is actually doing a clinical trial called the SMART clinical trial to generate data in the US for this broader patient population to, provide to the FDA to hopefully, I think the end goal would be not to speak for them, but to broaden the label in the US as well.
That's an interesting point, and we often get questions from people in other countries. If a gene therapy is approved in the US, does that automatically mean that it's approved in my country, whatever that country may be? No.
Separate process, and like we have in the US, the regulators could look at it slightly differently in terms of the prescribing label and who has access, the ability to access the drug.
So there's a potential that a clinical trial would have to be conducted in these other countries. And like you said, with the experience in SMA, it actually ended up with a different approval indication. Yeah. Now, in order for the U.S. to change for Zolgensma to be able to include individuals with that same weight requirement, is it another clinical trial that needs to be done?
And it just this is obviously a discussion that the sponsor has to have with the FDA and work out what the FDA would want to see. And I'm not privy to those conversations, but I can tell you that there is a clinical trial that was recently announced to look at patients with an expanded, a bigger weight, who are heavier and thus older, given that they're children. So.
And Dr. Brooks, recently there was a publication, and certainly it came through on many people's news feeds, about a clinical trial for a condition for AADC enzyme deficiency. And they did a gene replacement therapy, and they had an age range that went up to, I think it was 4 to 9 years of age, people that were in that trial. Can you tell us, well, I know you were not, let's say, the clinical investigator in that trial itself, but I believe you're familiar with it. I was wondering if you could tell us a little bit about what was seen in that trial.
Yeah, I've had no involvement in that trial, but I remember seeing some of the early data presented at a gene therapy meeting actually many years ago, because some of the earliest work was done in Taiwan. And it's a kind of an interesting disease where essentially AADC is necessary to make a neurotransmitter in the brain called dopamine, and to have the right levels of dopamine. And you need dopamine essentially for voluntary movement. And so if you don't have dopamine, you really can't move very much. And so the thing about AADC deficiency is that they need to target cells in a particular part of the brain. So they use this direct injection into the brain. It's called the striatum, I believe. And the other good news about AADC deficiency is it's not really a neurodegenerative disease. So the brain cells that would normally make AADC were still in the brain, and they still largely functional. They just lack this one enzyme to make this neurotransmitter. They can make do all the other steps. And so when the trial was done, the vector delivered the AADC gene into the brain, into those cells. They began to make dopamine, and that had a fairly rapid change in the way the patients behaved, because now you have dopamine, now they could move, and the results are quite striking to see. And I think it's an illustration, though, of that, in principle, the way we kind of think of this is monogenic diseases. You're missing a normal copy of the gene, and if you put it in to the right cells at the right time of development or in the disease progression, and that's important. If you do that, something good should happen. But what that something good is really depends on the specific biology of the disease. And this one is a kind of a unique situation where you got this rather striking response in the patients. But it kind of makes sense if you think about that it was not a neurodegenerative disease, and they're replacing this enzyme, and now they can make dopamine that has the ability to work also to some extent outside of cells. So in some cases, and presumably in Rett syndrome, you would get the therapeutic gene in the neuron or the brain cell, and then make the MECP2 protein, and that helps that cell and only that cell. But there are other conditions where you might do a gene therapy, and now you're making an enzyme that can not only help the one cell, but go to other cells. And you get this sort of amplification effect that you have in some of the lysosomal storage disorders, for example. And that was kind of the case in AADC deficiency.
Excellent. And is there any way to predict, before you've done your human trials, is there a way to predict what will improve in an individual once they get gene therapy?
No. I think the short answer is no. Jill?
We do our mouse studies or any of that. That doesn't tell you what your loved one with Rett syndrome will regain or maybe be able to do down the road.
I mean, in the mouse models in SMA, you're essentially looking at survival. Are the mice surviving or not, and how that will extrapolate to any individual given symptom in SMA, you know, motor function versus respiratory function versus swallow, you know, I mean, I don't think you can really, it's not that finely tuned. So.
Yeah, I think the mouse studies in most of these will give you a sense of what might happen in humans, but I don't think there's any way to really predict it until you actually test it, because a lot of these things seem fairly simple, like when we draw them up in diagrams, but the human body is very complicated and things can happen that we just don't understand. So I think, you know, there's usually animal data and there's good reason to expect that there could be some benefit. Otherwise, you probably wouldn't get an IND, but to really predict what's going to happen, I don't think anybody would want to do that with any level of precision. I wouldn't, at least.
Yeah. Yeah, and Dr. Jarecki, you may be able to speak to the SMA experience, you know, and I don't know if either of you know the details of the AADC enzyme gene replacement therapy, but was the response uniform, meaning that everyone that got the gene therapy all gained kind of the same skills, and was the timing of it uniform kind of across those that have been studied and now that are receiving it?
Yeah, so, you know, I went back and I looked at some data from this first clinical trial, which was the 15 patients in the phase 1 and there's been a long-term follow-up study of them just published last month in JAMA Neurology. And they had 10 patients from the higher cohort that went into the long-term follow-up study. And if you remember, SMA, these babies all had 2 copies of SMN2, so the severe form of SMA. The natural history would have them, you know, 75% of them being on permanent ventilation by, say, 13 or 14 months, none of them sitting. And at the 5-year follow-up, you have 2 of those babies walking, 2 of them standing without support, and 6 of them sitting independently. And that changes their SMA type from the severe form to the intermediate form, because not sitting was the definition of the disease. So right then and there, you can see that they're in the symptomatic babies. There's a spectrum of response. There are certain trends there, which is dosing earlier and having a higher baseline motor function, that the babies respond better. But there's a wide range of when these different babies gain the ability to sit. Some of them gain the ability to sit at 10 months, which would be in typical developing norms, and some of them gain the ability to sit at 32 months, which is obviously outside of the norm of a typically developing child. So a range of response. Now, SMA, of course, we already talked about as a neurodegenerative disease. And if you look at the pre-symptomatic studies where they were treated under 42 days of, within 42 days of birth, with the 3 copy babies, which would predict would be the intermediate form, 15 out of the 15 babies are within typically developing age norms. And then with the more severe form, the 2 copy babies, 9 out of 14 are within typically developing age norms. So, you know, pre-symptomatic treatment clearly is beneficial in SMA, but even there, it's not curative, you know, in these severe babies. We still have 5 of them that have a much milder SMA phenotype, but still an SMA phenotype. So.
Yeah, I might just add, I think if you look at some of the findings from the SMA trial, they're nothing short of, you know, spectacular for some patients who were treated at the right time. And others, as Jill just said, were not quite that way. And AADC deficiency, the results are also fairly striking, and the retinal disease, for which the retinal gene therapy is approved, I'm blank on the name of the disease, those can be pretty striking. But you would not want to assume that it always works really well, because there are other diseases, gene therapy has been tried that has not been particularly effective. And so it really is very difficult to predict. And you can also get, you know, toxic effects that were not predicted in animal studies. In some cases, and tragically in one of the more recent, about a year ago, gene therapy, some of those resulted in death apparently because of a liver abnormality that these patients had. That's a minor phenotype of the disease. It's a muscle disease. And many of the patients really had a great response, but some of them had this bad liver response, apparently because of this, you know, otherwise mild liver condition, and nobody ever predicted that. So I think it's very difficult to predict what's for one individual, much less a whole trial, whether it's going to work or not.
Thank you both for sharing that. And I think it's important because certainly in the Rett community, people are very hopeful for, Dr. Jarecki used the term curative. You know, certainly people are hopeful for a cure for their loved ones. And there's a belief that because of animal studies that were done that demonstrated you could get reversal of the phenotype even in a late stage mouse model, that if we can somehow just fix this gene, it could cure Rett syndrome. But I don't want to put words in your mouth, but it sounds like you do not feel, you would not call gene replacement therapy a cure. Certainly remarkable. I think that's the word you used, Dr. Brooks. But do you agree with that? You know, kind of, can you talk a little bit about that?
So I think there is, in our youngest patients, there is a remarkable change in natural history in SMA. But curative, I think, particularly there is some data in SMA that there, in the severe 2-copy form, a prenatal requirement for SMN, and this may be why we don't see full recovery in those patients. You know, a lot of this is biology and disease specific, so every disease will be a little bit different. But I would also say that there are certain stress points in patients with SMA, and it'll, you know, where patients with mild disease begin to have more symptoms. One of those would be around puberty, and that would be, I would say, because you have rapid growth and weight gain then, and if you have fewer motor neurons that are covering a much larger area than they normally cover, that's pretty stressful when they have to cover even larger areas, you know? So none of these children have reached these developmental time points where we can see what happens for them. That's why once you have gene therapy, it's really important that you have real world evidence tracking and you understand the change in the phenotype and with time, and you can think about what kind of, patients are still continuing to get the appropriate, clinical care, and you're developing new standards of care for the new presentation of the disease. So we just don't know what's going to happen in the future, and we have to carefully track that.
Thank you. And there's been a question from the audience. For those that received SMA gene therapy and now have gained some abilities, could they go back and get a different gene therapy? Because you mentioned it was clinical trial with something different. So we touched on this a little bit, but if it's not a redose of the same one, can they go get a different one?
Well, so patients with SMA very much want to do that. And the issue really is we have no data to show that adding 2 of these SMN enhancing therapies together will add additional benefit. So insurance companies will not cover that cost right now. There is a clinical trial called RESPOND right now, which is being done by Biogen, where patients who receive gene therapy, Zolgensma commercially, can come into a clinical trial and receive Spinraza, the antisense oligonucleotide therapy, within the clinical trial to see if there's added benefit. The clinical investigator needs to deem that the response to Zolgensma was incomplete, which, is a little vague. But yes, there is a great interest from our community to do that. But these are very expensive drugs, and insurance companies will not pay for both of them unless there is actual data indicating that the combination together is more beneficial than either drug alone. So clinical trials need to be done.
Right, and for those listening, so the Zolgensma is the gene replacement therapy, and then the Spinraza was, you mentioned it's an antisense oligonucleotide, so that is not a gene replacement therapy. That is a different type of therapeutic.
We also have a small molecule of Risti, which was recently approved, and of course, you can imagine combinations with that, so.
Yeah. People also have questions about cost. So what is the cost to families for, who receive gene therapy outside of a clinical trial? So once you have an approval for a gene therapy.
Well, in the US, I would say that I don't have the data in front of me. At CureSMA, we've done a lot of work to try to educate insurance companies and reach out to insurance companies who are not offering the drug according to the FDA label and try to put other restrictions upon it. But in general, I would say most patients have the ability to access the drug either through private insurance, Medicaid, or patient assistance programs.
And hopefully with the work that NIH is doing, ultimately the cost of these come down in the future for for disorders that are able to benefit from the work that NIH is doing. There's a question that came through about, I'm sorry, I just lost the question. Dr. Brooks, this is for you.
Okay.
There was a question about when we were talking about some of the preclinical work. Someone wanted to know, would studies with primates be required before a gene therapy clinical trial can be initiated, or is a mouse study sufficient? And maybe there's not a blanket answer to this, but
Yeah,
your experience.
I don't think there is. I think, and ultimately that always is a decision that the FDA makes. I think there may be situations, and we're anticipating this will be the case with ours, where we'll just be able to do mouse studies. But I think part of the reason, if that turns out to be the case, will be because we're using a vector called AAV9 that's been used in a lot of other studies. And route of delivery that's been used by others. So there's a certain familiarity with the safety profile of AAV9 in general. But my guess is if somebody came in with a brand new vector, there'd be greater likelihood that there would be a need for non-human primate studies. That's my guess as someone who doesn't work for the FDA, and I would not want to speak for them, but that's my understanding.
Yeah. Great. And Dr. Jarecki, there was a question for SMA. The question was, in Rett syndrome, there's a concern about too much of the gene, MECP2, because we have a syndrome called MECP2 duplication syndrome. And so there's this, mantra that you need just the right amount of MECP2. Is that the case with SMA?
So by genetic means, people have increased SMN protein levels to very, very high levels in mouse models, and there's never been a finding of a toxic response to very high levels of SMN. Recently, there was a paper published looking at gene therapy delivery of SMN in a mouse model, but where they did see some issues specifically in a particular type of neuron in older mice. However, that used a different promoter, a different delivery system, a different vector entirely than our approved gene therapy. So it's very, very hard to interpret those results, but increasing SMN by genetic means is not shown to have a toxicity issue in SMA. But that's clearly something that will be a concern in other, could be of a concern in other diseases.
Absolutely. Good. And Dr. Brooks, there was a question that you answered early on. I think someone jumped on after, but what are the conditions that are being looked at in the PAVE-GT pilot project?
Yeah, so there's two metabolic diseases, propionic acidemia and methylmalonic acidemia, but a particular form caused by a mutation in basically a very, very rare form of an otherwise very rare metabolic disease. And then the neuromuscular diseases are CoQ deficiency and DOX7 deficiency.
Thank you. Yeah, the question also wanted to know if Rett syndrome was one of them, so I wanted to make sure that we answered that. That Rett syndrome is not one of the selected conditions, and you talked a little bit about why you selected those four earlier in the conversation.
Mm-hmm. Yeah.
Another question that came through is there a possibility that using a gene replacement therapy could cause changes to something that was not intended to happen? So kind of what we might call an off-target effect, or is there a possibility of that? Would either of you like to respond to that?
I'm not quite sure what that means, an off-target effect.
Well, they say, is there any chance using this technology that it could change something that was not the target? So you were trying to replace, in Rett syndrome, MECP2, but could you accidentally do something else?
I don't think so. I think that what is getting at there is perhaps the concern that really has to do with genome editing, where you're going to try to edit and correct a specific region of a gene. And there is the possibility of what are called off-target effects, where you end up editing and changing the sequence of a different gene. And that's certainly a concern that has to be addressed in genome editing. But for gene replacement therapy, I'm not exactly sure how that would be a problem, unless I can't quite see that unless you got the gene expressed in the cell where it wasn't supposed to be. But there are ways to to limit that. So, Jill, what do you think?
I mean, I guess that's the question. If you express it in a cell where it isn't normally expressed, could that have some consequence? But I mean, that's the place you could think about that.
Yeah, I mean, you
And
hope that you would address that with animal studies.
Sorry, Dr. Brooks. I think you made a good distinction about genome editing, where you're trying to correct something, and the gene replacement. And I think the visual you showed earlier, where you're kind of putting your gene of interest in a little box and it's being delivered, and it's the gene, or in some cases maybe a mini gene, but you're not kind of cutting out and trying to correct one little part of it. You're saying here's a functional copy for the cell that you've targeted.
Yeah, and the non-functional copy is still there and still doing whatever it did. It just doesn't, it sort of doesn't matter at that point. Whereas in gene editing, you deliver an enzyme that goes and cuts the DNA sequence and changes it back to the wild type. And that gene therapy and gene editing sound the same in some ways, and the outcome can be the same, but the details are actually very different. And at this point, gene therapy is much farther along in terms of going into the clinic than gene editing, particularly for diseases of the CNS.
Excellent, thank you for that. One additional question I wanted to ask, people have wondered, and this came up particularly when the COVID vaccines were first rolling out, because people hear about AAV, and in particular, one of the COVID vaccines was using adenovirus. Does having the COVID vaccine mean that you can't get gene therapy, or if you've had this gene replacement therapy, does it mean that you shouldn't get the COVID vaccine? Is there any connection?
No, I think the way I'll try to answer that is that I think adenovirus and adeno-associated virus, they sound the same, but they're very, very different. So adeno-associated virus was called that because it was discovered by people who are trying to purify adenovirus, and they kept coming up with this contaminant. They just called it adeno-associated virus. But adenovirus is a human pathogen. If you get infected with it, and many of us have, it causes a pretty severe cold that you then get rid of if you have a functioning immune system. And it can be used for gene therapy, but it causes a much greater immune response, which is partly why it's used for vaccines. But it shouldn't. But having that vaccine, the one that's based on adenovirus, would have no effect on your ability to get AAV gene therapy.
We have this question a lot in the SMA community, given we have an approved AAV9 therapy, and we believe the same thing. It has no interaction.
Thank you both for answering that. All right, well, I know we're coming up to the end. So, many of you have submitted either through pre-registration or today kind of questions that are very specific to gene therapy and Rett syndrome. And really this topic was about gene therapy in rare diseases, which Rett is one as well, but there's still so many unknowns in Rett syndrome because we haven't had a clinical trial yet, so we don't have the experience to know. What we do know is that there are two companies that I mentioned earlier today who are working to develop clinical trials for Rett syndrome. And both of these companies, as I said, have announced that they're going to submit to the FDA their investigational new drug application, or IND, by the end of 2021. And this application contains the lab studies that have been conducted on the safety of the proposed therapy, any prior experiences in humans, which there isn't for this case, information about the manufacturing, as well as their proposed clinical trial design or their clinical protocol. The FDA may require them to make changes to that. So until the FDA gives them the okay, we don't have answers to a lot of the questions that people are asking. But once any study is approved on the FDA side and goes into clinicaltrials.gov, we share it out on our Rett Research Ready My Rett Trial Finder so that the families can learn about it right away. You know, the details we know. There are these two companies. One company has a regulatory element that's intended to provide feedback loop based on the amount of MECP2 that's in the cells. What we do know is, as we talked about earlier, with the technology today, gene replacement therapy is a one and done, meaning you get one dose as of, you know, what we have today. And Dr. Brooks mentioned, it also means that with today's technology, someone who's had AAV9 delivery of gene therapy could not necessarily participate in another trial. So I said there's two different companies that want to conduct clinical trials. You can't really do both. As of right now, you can do one. Details that, you know, we don't know are the rest of the details. We don't know what the expected outcome will be. Will a child with Rett syndrome verbally communicate, walk, use their hands like they're typically developing age-matched peers? Will results be better or worse than what was seen in SMA, which is a question that has come up? How long will it take to see these changes? Is there an age group that will be more or less responsive to this type of therapy in Rett syndrome? Will both males and females with Rett syndrome benefit from this therapy? Will clinical trials include males and females? Will the clinical trials start soon? We don't know when they're going to start, assuming that FDA ultimately gives them a green light. We also don't know, as I mentioned, who will be eligible for those clinical trials until the FDA gives that final approval. We don't know if gene replacement therapy works in the clinical trial, how long it'll take to be available on the market for the community. So that time, Dr. Jarecki talked about the first trial was in 2014, and FDA approval came in 2019. That was a 5-year time period. We don't know what that time period would be should it be something that works. We don't know if using it in combination with a small molecule would have added benefit. All of this is stuff that we need to learn about. But this is really an exciting time for Rett syndrome. There's so many different approaches being studied. Gene replacement therapy is one of them. And really our role at the foundation is to help to provide that education so that you can feel empowered with the information so that when it's your turn to think about, is this a therapy that you want to consider for your loved one? Should they be eligible? That you feel like you've started to answer some of these questions beforehand and had time to think through. So again, I want to thank Dr. Brooks and Dr. Jarecki for joining us today. It was great to learn from your experiences and from your knowledge in this space. And thank you for sharing this with the Rett community.
Thank you.
Thank you. Happy to help.
All right. And with that, we are going to close this out. Paige, I do want to share, I want to see if we can share out the American Society of Gene and Cell Therapy patient education page, maybe with a follow-up link, because I think that's a really great resource for families to start looking at, to learn in general about gene therapy and some of the questions you might want to be asking. All right, so thank you very much, everyone, for tuning in, and we hope you have a great rest of your day.
Thank you, everyone. Thank you, Dr. Brooks. Thank you, Dr. Jarecki. Thank you, Dr. Pichard. This information was incredible, and I hope it empowers everybody in ways great and small as you move forward in your day-to-day life. We know that the days with Rett syndrome can be very long, but we know that they will be better when you're empowered with information. So you'll receive a link to this recording soon, replay it, digest the information, let us know if you have more questions. We will certainly bring further RettEds to you. Take a moment to answer the survey at the end of this RettEd because you'll have an opportunity to ask for future RettEd topics that we can bring to you to help you in your day-to-day life and keep the hope for research and better treatments and intervention alive because they are coming. Thank you for all of the work that you do. We wish you both good luck and much success in your continued work in rare diseases. And we will see you again for future RettEds, and we hope to see you all in Nashville in April. Thanks for being with us today, and we'll talk to you again soon. Bye-bye.
Thank you.